All right, so why don't we go ahead and get started here in the interest of time. It's a couple minutes after. So thanks everyone for joining us today. We at ISTC are extremely excited to be releasing our 2020 R&D Index, as well as our third annual Researchers to Know today. I'm Matt Bragg, Director of ISTC. CEO, Colleen Egan, who has, am I still here? I was muted for a second. We'll throw it over to Colleen now for an intro and welcome. Oh no, you're on, you're on mute there. Edith, can you make sure she's unmuted?
How's that?
There we go.
Yeah, I don't know what happened there. I was unmuted the whole time, but sorry about that. You saw my mouth moving, but unless you could lip read, you couldn't figure out what I was saying. But I was welcoming everyone. Good afternoon, and welcome to the Illinois Science and Technology's 2020 R&D Innovation Index release and our third annual Researchers to Know event, as Matt said. We're thrilled to have all of you here with us today. And for those of you who don't know me, my name is Colleen Egan, and I'm the new CEO of ISTC and our K-12 educational partner, the Illinois Science and Technology Institute. I started my career as a nurse. I moved into administration, and just over 20 years ago, I focused my career in technology. By 2018, I was named the Illinois Technology Association's Prominent Woman in Tech, not only for my professional work, but for my dedication to STEM, to diversity, equity, inclusion, gender equity, and educational equity. As a serial entrepreneur and a C-level executive, I've had the great privilege and opportunity to personally and professionally explore and innovate my life, my career, even now. I was interviewed and hired through this COVID-19 pandemic. And in the last 7 months, I have seen more inspiration, motivation, and innovation than at any other time. The world may have stopped in numerous ways, but innovation has not, and we have the great honor of presenting some of that innovation here today. I'd like to thank our presenting sponsor, Sterling Bay, and all of our index data partners. Our work would not be possible without all of you. I'm also fortunate to have an amazing team, and I'd like to give a big thank you and shout out to Matt Bragg. He's the Coalition's director, and I'd like to thank him for his dedication and the countless hours needed to put the index and this event together. I'd also like to thank Edith Portales for supporting us every day in our work. Her thoughtfulness and guidance is invaluable to the team. So once again, I thank you for being here today with all of us. I thank you for your continued support of ISTC. And now I'd like to pass it back over to my colleague, Matt Bragg.
Well, thank you, Colleen. I appreciate the kind words. Next up is Dr. James Gillespie from Sterling Bay, Executive Director of the PRISM Institute. So over to you, Dr. Gillespie.
Great, thank you, Matt. And a pleasure to be speaking to this great group at this distinguished conference. Thank you, Colleen, for being such an inspirational leader. The thing I want to note at the top is that we are looking for collaborators. So I'm going to talk about the really wonderful space that's going to be arising on the north side of Chicago, but we are actively seeking university collaborators, so feel free to reach out to me directly. I'm James Gillespie. I'm Director of Scientific Strategies at Sterling Bay, as well as I lead the PRISM Institute. And I have 21 slides, but mercifully for you, Colleen has said 5 minutes, and then I go on mute. So next slide. So Sterling Bay, we are all about building. We love to build buildings, but really for us it's building trust, relationships, and ultimately building value to our partners and then communities. Next. So we have a great track record. We've done some great iconic buildings here in Chicago, here are a couple there, and even one in Oregon. Next. And then we've also done some really great work in terms of building corporate headquarters. Here shortly you'll see a picture of the great McDonald's headquarters in the West Loop that we built. as well as the Google headquarters for Chicago that we built not far from there. And then next. Sterling Bay, though, and even though we're based in Chicago, we have a national footprint. So on the next slide, you'll see we're actively doing projects in Portland, Miami, and Dallas. And we have just announced the Sterling Bay West. So we're actually going to be starting operations in Los Angeles in the Bay Area, and eventually we're going to go to the East Coast. But Chicago remains our base. We all love the city. We all live here, work here, and it's just a great city in terms of culture, diversity, talent. And by many measures, it's a top diverse economy in the U.S. Next. The real strength, and everyone on at this conference will appreciate this, are our great universities here. And again, we want to collaborate with you. In the aggregate, these 11 terrific universities in our state do over $2.5 billion in R&D research. So Sterling Bay increasingly moves into life sciences. We see you guys, you folks, as important collaboration partners. Next. So Sterling Bay has already had some successes in healthcare life sciences. The next building here is 2430 North Halstead, which is a life science lab, about 120,000 square foot, already filling up with some great tenants. Next. And then there is 600 West Chicago, the old Montgomery Ward building. And that has also some great healthcare and life science tenants. Next. But the thing we're really excited about is Sterling is Lincoln Yards. It's going to be a 55-acre development in the north side of Chicago that we're building from scratch. So it's going to be a really cool space. You can see these really great pictures capture some of the flavor of it. And it's going to include, though, 21 acres of open green space. Next. And the next slide you'll see is a satellite view of it. And you can see that Lincoln Yards sits at this intersection of these really cool, iconic neighborhoods, Lincoln Park, Bucktown, Wicker Park. And we're just, of course, those of you all know Chicago's geography, just 10 minutes from Chicago, and we sit right along the Chicago River. Actually, as you'll see in the next slide, the next one, Matt, we actually straddle the Chicago River. So it's a really exciting project. There it is there, the 55 acres. And it's going to include 100 shops and retail spaces, or 5,000 residences and 1,000 hotel rooms. Here really quickly, there's some visuals of the building. You can see it's going to have a great view of downtown Chicago right on the river. Next. The next view gives a nice close-up of some of the tremendous decks that are going to be there. And the balconies. I'm not an architecture real estate person, but I think we can all appreciate, a really cool building when we see it. And this is going to be that. Next. The next shot just is an interior atrium. You can see that this is not just going to be kind of a lab space, dark. This is ultimately going to be a space to collaborate and work and network. So we're really excited about what the interior is going to look like. Next. We just have a quick view of one of the floor plans. The thing that the note here is that we're really going to emphasize the merging of the lab and the office space, and we believe that's conducive to collaboration. And then we can go to the next one. And then the next 3 slides, the key part is that the key part of these next 3 slides is that we're situated in the context of a really cool 55-acre neighborhood we're building from scratch. I was at MIT undergraduate at MIT. When I was there, we were just starting Kendall Square. And so we want to have kind of that flavor here where it's live, work, play, grow. And the next, because scientists also like to have fun, right? Researchers like to have fun, we're going to have some great entertainment spots. The next, also some really cool open plazas. And then we'll go to the next slide. I know there's a little lag here. We'll get to that. And so you can see that, ice skating, et cetera. And then finally, the last substantive slide is we are going to have this great open park space, because scientists also like to get out and get some fresh air and walk around. And so we're really excited that about 40% of this space is going to remain open green land. So I know I went through that very quickly, want to keep us on schedule, but do feel free to reach out to me. We love to talk to you about the space. And I'll say one final time, we are actively looking for university collaborators. Thank you, Colleen. Matt.
Thank you, Dr. Gillespie. Great. So with that, thanks again to Dr. Gillespie and Sterling Bay for supporting the index and researchers to know this year. But with that, we will jump into a little bit of the new data before we get to our researchers.
Can I add one thing, Matt, before we start?
Of course.
I see a lot of chat in the chat box looking for your presentation, Dr. Gillespie. So if you would share your email, maybe people could reach out to you directly.
Yeah, and we can also include that information.
We'd be glad to do that.
Great.
Yeah, we can include that in a follow-up email after the event as well.
Perfect. Thanks, Matt.
Yep. Okay, so for those of you that might be less familiar with ISTC and who we are, we're really an economic development nonprofit at our core. So we exist to help grow strong jobs in the state through support of science and technology, innovation, and entrepreneurship. So we like to say we measure, connect, and advocate for the state's innovation economy. We've been around for more than 30 years now. We were originally started by the state of Illinois as kind of a tech advisor and economic development partner. Today we're based at 1871 when we're able to be in the office in person. We act as a shared voice for the state's R&D community. So today our work really falls into three buckets. So data, which we're going to talk about today, but then also advocacy and education through our institute. So on the advocacy side, at the state level, again, we act as a voice, a shared voice for our members, and really, represent our community to state lawmakers and advocate for pro-innovation and economic growth policy. And then on the federal side, we again translate the needs of our community to our federal delegation. And then, really a founding mission of ours is to go to the federal level and try to bring money into the state for these economic development initiatives around science and technology. I would be remiss in all this not to mention the work of our great institutes. The STEM Challenges and Mentor Matching Engine are the two main programs that the institute runs, connecting high school students with professional mentors at companies across the state of Illinois. So the STEM Challenge program is an academic year-long engagement between high school students and mentors at companies in the state. And that program leverages the Mentor Matching Engine, which is our all-in-one virtual mentor solution that connects these mentors virtually to students, which obviously right now is more important than ever to keep creating those connections virtually when so much of our education has unfortunately moved online. So if you're interested and want to learn more about ISTI, I'd encourage everyone to check out their Facebook page, which is facebook.com/the-ISTI. And they have some really great interviews up there that they've done recently as well. So today we're gathered to talk about the Illinois Innovation Index, which is our data-driven report that comes out 3 times a year. We do one in the spring on university entrepreneurship, where we track all of the startups coming out of our universities, as well as kind of the tech transfer and commercialization activity going on at universities, and track things like, what happens to these companies when they leave campuses? Are they staying in Illinois? Are they able to raise money in the state? That kind of thing. Next is our R&D issue, which we're obviously going to talk about today. And then lastly, which comes out in the winter, is our STEM talent issue, which really tracks, STEM degrees coming out of our universities, talent migration, so who's coming into and out of the state, as well as demand for those jobs, so the workforce for STEM in Illinois. Okay, so as I mentioned, we're all here for the R&D Index. This is a report that we're really excited about because it really tracks at its core innovation activity that's happening in Illinois. I think when you look at the 3 issues of the index that we put out, R&D is maybe the most nebulous as to, why do we track this activity? Why is it important? And what we like to say is that R&D is really the lifeblood of innovation, right? So companies use R&D to create new products and services that keep them competitive and grow jobs in the state. Our universities use research to expand our knowledge of basic science and are increasingly commercializing new technologies. And then our 2 great federal labs really use R&D to keep us on the forefront of discovery and kind of ahead of the technology curve. So we track R&D activity across businesses, universities, and federal labs, and put that all together to look at total R&D. What's new in 2020 is we really took an input and output view of this analysis. So really assessing the resources invested in R&D and then the resulting output from that. So with that, we can jump into some of the data itself. So in 2017, which is the most recent data year that we have for all of this activity, the state produced 18.1 billion in total R&D activity, and that was good for 7th nationally. We did see a pretty significant uptick in total R&D activity in 2017. It increased by about 600 million from 2016 to 2017. But despite that 600 million uptick, we have seen total R&D activity in the state increase a little bit more slowly than it has nationally. So as a result, you'll see that we've kind of dropped a couple of spots as we go through some of this data. So we'll start with business R&D as we dive into those three kind of pillars of R&D happening in the state. So looking at both the inputs and outputs here, 14.4 billion in expenditures by businesses on R&D, which is good for 8th nationally. And then over 260,000 science and engineering employees. So these are the folks that are, I think, most likely to be in R&D roles. And we have the 7th largest sort of R&D workforce by that indicator. And then on the output side, so we've had more than 7,000 patents awarded to businesses in 2019, which is 7th nationally. And then 745 billion in private industry output, which you can think is kind of GDP specifically for businesses. And that's good for 5th in the nation. So looking a little bit deeper at expenditures, again, the state ranks 8th with 14.4 billion. And that activity increased by about 660 million from 2016 to 2017. But overall, business R&D expenditures since 2013, unfortunately, have grown at about half the rate of the national average. So diving into industries, so where are we strong in R&D activity? Where do we maybe have room to grow? What we see here is that pharmaceutical R&D really kind of dominates, right? It's 3 times more in terms of business R&D expenditure than any other industry in the state. And we rank 5th nationally in R&D and pharma at the business level. But the state is also a national leader in machinery R&D, food R&D, and finance and insurance, which I think really makes sense when you think about the landscape of companies in the state. Machinery, so you've got CAT, you've got Deere, you've got ITW. In food, we have some of the largest food companies in the nation with ADM, US Food, Ingredion, Kraft Heinz, a lot of companies in food. And then finance and insurance. The state is really a leader in finance and insurance with Allstate and State Farm and Discover and others in the state. So growth among these key industries. So unfortunately, we have seen some of these really leading industries kind of lag national growth trends. So for example, in pharma and machinery and computer and electronics, the growth rate for R&D in those industries just hasn't been as strong as it has been nationally. But luckily, we have a few industries that have really accelerated growth in the last 5 years. So R&D in IT and software, in food and medical equipment has all seen growth at or near 20% annually since 2013. So those are industries that are really growing quickly in the state. So unfortunately, we don't have a lot of data at the company level about R&D. It's something that, as competitive businesses, they're not very willing to share data on, the R&D that they're working on for competitive reasons. But one area that we do have business level data on are patents. So again, over 7,000 patents in 2019, which is 7th most nationally. And when you look at some of the leading patent producing companies in the States, You know, it's kind of a who's who of pillars of industry in Illinois, right? You've got companies like CAT and Motorola, State Farm, Allstate, but you also have companies that are based elsewhere but have large R&D presences in Illinois. So like IBM and JP Morgan are a couple that are near the top here. Okay, so moving on to university research. So as Dr. Gillespie actually mentioned, and I think he used our data for this, we've got more than 2.5 billion annually in research funding to our universities in the state, which is 10th nationally. And then in terms of R&D personnel, so you can think of these folks as PIs, or those that are getting paid through research budgets. Nearly 40,000 R&D personnel in the state, which is 7th nationally. And then 6th nationally in square footage for research. So about 9.2 million square feet of research space in the state at our universities. And then in terms of output, right? So funding, personnel, space, those are all inputs to this process. Outputs are academic publications, patents, and then licensing income are the three that we've identified here. So over 13,000 publications, which is good for 6 nationally. We're 9th in patents with over 250 a year. And then we're actually 4th in licensing income. So this is really an area where the state has excelled in, you know, when new technologies are created on university campuses, we're able to license those and the market really values them. We're at almost 300 million a year in terms of gross licensing income from those activities. And when you look at sort of the inputs and outputs here, I think what the takeaway is, you know, research funding has lagged a little bit, but when you look at the outputs, you know, 6th nationally in publications, 4th in licensing income, I think the takeaway for me is that, you know, the state's universities are doing more with less in terms of turning, you know, fewer research dollars into more really tangible outputs. So diving deeper into that research funding, again, almost 2.6 billion in 2018. We ticked up about almost 70 million between 2017 and 2018. But overall, over the last 5 years, unfortunately, again, that funding just hasn't quite grown as quickly as the national average. And as a result, we've fallen actually from 8th in the country in university research funding to 10th. So looking at this research funding, excuse me, by field, you'll see that research activity in life sciences is actually, there's more research funding in life sciences than all other fields combined, more than 1.5 billion in 2018. Looking at some of the state's other leading fields, we're 8th in life sciences, 11th in engineering, 8th in physical science, and then 8th in computer science. And then by source, so, you know, where is all this money coming from, obviously, for research? Like most states, most of our university research funding comes from federal agencies. But we are leaders in several of those in receiving funding from those agencies. So we're 3rd in funding from DOE, which is thanks in large part to research going on at our federal labs, which kind of ties in with several of our universities in the state. We're 5th in funding from NSF, and the University of Illinois is one that always ranks highly in NSF funding. We're 7th in funding from DOE, and then 8th in funding from HHS, which includes the National Institutes of Health. So, you know, universities like Northwestern, UChicago, UIC are all leaders in funding from NIH. So the last kind of input and output analysis I want to mention is university industry collaboration. And I'll talk a little bit more about this in a while. But essentially what these are, you know, co-developed R&D projects between universities and industry. So what these collaborations do is really leverage the strengths of industry and companies, right? So for universities, you know, working with a business gives them obviously increased research income, but also, you know, access to market insights and things like that. And then for businesses, obviously you're accessing world-class researchers, you're accessing, you know, research facilities that you might otherwise not have access to. So it's really, you know, leveraging the strengths of both sides to increase innovation. So in 2018, we were 8th in the country in university research funded by industry, and then 6th nationally in shared grants between university and industry. And then on the output side, in 2019, we were 8th in co-assigned patents to both the university and a member of industry, and then 10th in co-authored publications. So finally, research happening at our great federal labs. So each year, our federal labs, Argonne and Fermi, conduct more than $1 billion in R&D activity. And that research activity increased in 2018 by about 50 million for Argonne and almost 10 million for Fermi. And this is, I should point out, this is before the announcement of the 2 national quantum institutes, one at Argonne and one at Fermi. So we're expecting this activity to increase significantly over the next couple of years. I did also want to mention user facilities at both Argonne and Fermi that are really connecting the labs to industry and kind of allowing industry members to access some of the facilities at the labs and grow innovation in that way. Okay, so I've gone through a lot of data in a relatively short time, but I do want to leave everyone with a few takeaways before we get into our researcher presentations. So overall, some of the challenges, we do lag in R&D growth on the business side from some of our key industries. But luckily we have seen some other industries kind of pick up, accelerate their R&D activity. Overall university research funding, we just haven't seen the growth that I think we would all like to see. And luckily, I think universities are being really efficient with turning that funding into, quality research output. But of course, we would like to see that research funding increase. And then the third challenge is, the reality of where we are with our economy, right? And the unfortunate nature of coronavirus and what it's caused nationwide. So a key challenge is going to be how do we navigate this and keep up our R&D activity so we maintain kind of long-term innovation goals. On the strength side, as I mentioned, business R&D growing in several industries like food and IT and software being 2 key ones where business R&D is really accelerating in the States. And then second is, the synergy between life sciences at our universities and our pharmaceutical R&D industry here in the States. And I think we're already seeing collaborations and have for a while between some of the leading pharmaceutical companies in the States and our great life science research at universities. Third is our federal labs. And, these have been a staple of R&D in Illinois for a long time. But I think we're seeing them continue to lead in really key areas with the announcement of the 2 national quantum centers at both Argonne and Fermi. So that's, really exciting to see and encouraging to see them continue to lead in what's going to be, I think, a really transformative technology. Lastly, on the strength side, I do want to mention a few new collaborative initiatives. We just talked about kind of university industry collaborations and the importance of those. So new collaborations, things like the Discovery Partners Institute, the Illinois Innovation Network, the Chicago Quantum Exchange and their role in growing Chicago as a hub for quantum, and even things like the new Math and Statistical Institute that just got announced. That's a collaboration between several leading universities in the state. So looking forward, again, it's going to be important to continue to grow R&D activity during what's a period of economic uncertainty in the state. I think we really need to be intentional about making sure that we continue to grow this activity to ensure that we're not threatening the long-term innovation that's going on in the state. Again, supporting the growth of new collaborative R&D initiatives. So we've seen several of them pop up over the last 5 years. And I think now it's really about making sure that they're supported and have the resources they need to really drive innovation in Illinois. But then lastly, excuse me, I did want to mention creating new collaborations between businesses and universities in the state and really focusing on more discrete collaborative activity. So when we have, say, a business that's right in the backyard of a university, making sure that they're aware of the resources that exist within universities and encouraging them, if you're a business that wants to access the research expertise at your university in your backyard, that you're able to do that. So we've proposed a program called Illinois Innovation Vouchers that would make that possible and incentivize some of that activity moving forward in Illinois. So with that, I do want to give a special thanks to all of our members and partners and sponsors and everyone that came together to make this report possible. A big shout out to Sterling Bay for their support of this issue of the index, as well as our great data partners, including Wellspring, who was kind enough to give us access to their data platform, their Scout data platform, which is a really, really cool platform if you're interested in diving deeper into some of this activity. And then to all of our members and partners that support this research. Okay, so with that, I want to move on to our researchers to know in 2020. So we released this today on our website, so everyone can check out the full list, which includes biographies for each of our researchers, which is on our website at istcoalition.com/data. I won't read off everyone's name here. I encourage everyone to go check out the webpage. But we are fortunate enough to have 10 of our researchers on the call today to give, excuse me, a brief presentation on their research. So with that, I will jump to our first researcher, who is Vinayak Dravid from Northwestern University. So Vinayak, if you want to unmute yourself, and then I will advance the slides when you tell me to.
All right, before we begin, I just want to thank the organization, particularly Matt and Edith and others, for the opportunity to share our excitement with this community. I'm looking at the composition of this group and the last winners. I feel flattered and delighted to be part of this very special elite group. I've been in Chicago at Northwestern for almost 30 years. I have 2 boys who graduated from Northwestern and a wife who's also a Northwestern graduate. So we are deep purple. So I bring greetings and wishes from our president, Marty Shapiro, our dean, engineering dean, Julio Tino. And every time I go give blood twice a year, they complain. My blood always is very deep purple. So that's me for you. So my research program is about nanotechnology, as you can see. And I'm already giving you the answer that nanotechnology for environmental remediation is our program that we have developed the last few years. We believe it's hip, full of hope, and I don't think it is a hype. And below that are my affiliations. You're welcome to contact me. I want to just shout out a couple of things. One is the Nuance Center that I run. So one of the things that have changed in the university in the last couple of decades is not only do we produce fantastic students, which you are, and the scholarship that we do, a place like Northwestern or Urbana, but we also provide some infrastructure, nanotechnology infrastructure. So the Nuance Center and the SHINE resource is an NSF Center of Excellence in the Midwest. The first time ever that NSF has established a center for facility and infrastructure in the Midwest. And we're very proud to be the second renewal of that program. So any small industry, large industry, need access to nanotechnology tools and techniques, you're welcome to visit us and we can work with you. So the next slide is about, Matt, is a video clip. So do you have that video on the?
Yeah, let's see if we can pull it up.
It'll be great to give it as a backdrop. Yes, perfect.
All right, hopefully it's not too loud.
10 years ago, a powerful explosion destroyed an oil rig in the Gulf of Mexico. The Deepwater Horizon well dumped an estimated 168 million gallons of oil and 45 million gallons of natural gas into the ocean, making it the largest accidental marine spill in history. The event sparked a nationwide call for more research into oil spills and their impacts on our oceans, environment, and lives. Studies over the last decade have advanced our knowledge of oil spills, resulting in novel applications of emerging technologies. One brand new technology is Northwestern University's Smart Sponge. This highly porous sponge has a nanocomposite coating of magnetic nanostructures that attracts oil and resists water. The coating can be applied to any cheap household sponge and binds with oil molecules, capturing and storing the oil in the sponge until it is squeezed out. The Smart Sponge can absorb more than 30 times its weight in oil and can be reused up to several dozen times without harming marine life. The National Science Foundation funded team says right now the Smart Sponge can be used effectively to remove pollutants and toxins in water and soil. It can also clean up smaller oil spills in a more economical, efficient, and eco-friendly manner, helping to protect ecosystems and surrounding communities.
Thank you, Matt. So this gives a little bit of backdrop of, what started as a basic science. So I think, Matt, I enjoyed the earlier presentation about the resources at Illinois. In fact, this research program initially started as a biomedical imaging. So that's primarily the work that I do. And it's morphed into environmental remediation. So one such product that we've recently developed, for which we got a lot of traction, is the OHM sponge. It stands for, as you can see below, oleophilic. It loves oil. Hydrophobic. Whenever we are sucking up oil, we don't want water to contaminate. So it needs to be hydrophobic. That's H. And M is magnetic. So it allows us to move things around, and there are other benefits of being magnetic. So this OHM sponge is very economic because the material that we use are very eco-friendly, very cheap, iron and carbon based. And this sponge that you see here, you can clearly see on the left-hand side, there's a water droplet, so that repels the water, but the oil it sucks up, as you see in the video as well. So it's very efficient, almost 200 times its self-weight you can recover. The most important thing among others is this oil can be recovered. It doesn't get burnt or spread out in ocean or waterways. It actually can be recovered and then monetized. The green chemistry, reusable, and as you say, it's effective against all sorts of water mixture. So we're very excited to deploy this technology. In fact, just before this talk, I had a talk with US Coast Guard, and ICOPAR is a 15-agency consortium for oil pollution in America. And we were excited that they're very interested in using the sponge as a potential oil remediation approach. The next slide shows that this is just one example of environmental challenge that we face. So as the modern society evolves, we see microplastic, we see contaminants in water, and those are sort of a sideshow of societal development. So it's very important that we use technology to remedy these challenges. So we have developed, as I just showed you, OHM sponge for oil, but we have also developed membranes which can suck up toxic metals and excess nutrient, which is a major problem in Lake Erie and other waterways. This excess fertilizer creates algae bloom, and they look so ugly, and they suppress the sunlight going into the deep waters, and that causes toxicity as well. So we have developed this concept into nutrient and toxic metal removal. And the next slide shows the way this process works. We start with the commercial sponge. These are typically the ones you tend to discard. So when you get your gift from Amazon, your item of interest may be small, but most of the packaging is waste packaging, the sponge. All we do is take the sponge, mix it in our coating. So only 3 to 5% of our coating makes it a smart sponge. And then you can use air, soil, or water as your sample, and it absorbs, as you see, with oil or nutrients. You recover them and then reuse the sponge. So you can do this circular economy. And so there is no waste material because everything gets used. And what you see on the bottom left are the 2 courts, which I often do. This is a week we're starting to teach. And I tell my students, if you look far ahead, then when you see it looks like a science fiction, maybe you're wrong. But what you should do is should look like a science fiction. Otherwise, it is definitely wrong. So we are asking our student to be aspiring and ambitious in their approach. And this is one such example that we're very excited to share with you is nanotechnology. So it's a nanoscale solution to a gigaton problem. And with that, I think the next slide is, this is it. So with that, I thank you for your attention. As I said, I bring greetings from my research group and look forward to working with some of you. We have a startup company which we want to keep it in the Chicago area. That's one of the challenges in Chicago is you get attracted to go on the East Coast or the West Coast. But our MFNS startup, we're very committed to stay in Chicago, and I look forward to working with some of you. Thank you very much, Matt and Edith, and I appreciate the opportunity to share with you.
Great. Thank you. Yeah, thank you, Professor Dravid, and definitely need to keep that startup in Chicago. So, yeah. So next up is Professor Elizabeth Gaillard from Northern Illinois University.
Okay, can you hear me all right?
Yes.
I'll put my video on just for a second, long enough to wave hello to everyone. Nice, thank you.
Hello.
I just wanted to thank the organizers again for inviting me to participate. This is a fantastic opportunity. I'm sorry that I have to follow that beautiful video, but somebody had to be second, right? So I've been a professor at Northern Illinois for a little over 20 years now, and have really watched this university grow and become a fun place to be. I have a longstanding interest in the biochemistry of eye diseases, particularly ones that are the result of the natural process of aging. So in the lower left, I've just put some faces of people moving through transitions in life. And I wanted to include that because I think we all sort of intuitively know that the eyes are something that we always focus on. So not to make a pun, but the eye is a very, very interesting organ. Some of the things that are particularly interesting about it contains the most and least metabolically active tissue in the body. It has its own sort of unusual immune response that is of particular interest to me. And it's a very highly compartmentalized organ. The two major diseases that we have studied in my research group over the years are age-related macular degeneration and diabetic retinopathy, both of which are now competing to be the leading causes of blindness among older persons in industrialized nations. And the graphs on the right show you that both of these diseases are projected to continue to increase very rapidly over the next 30, 40 years. So we have, if you'll go to the next slide, please. My research group, we primarily focus on very basic fundamental research, but it turns out that some patentable technologies arise from this type of research. So let me give you an example of that connection between basic research and intellectual property. One thing that connects both age-related macular degeneration and diabetic retinopathy is the eye experiences the growth of many new blood vessels. These blood vessels are weak, they tend to leak, and they stimulate a very strong inflammation response within the eye. Currently, the only way to treat this issue is to give people direct injections of a drug that slows down the growth of new blood vessels. So this is one of the interesting things about the eye is it's able to clear itself of materials that are in it. So the injections have to be repeated about every 4 weeks. They're very, very expensive. And anytime you make an injection into the eye, there's an opportunity for infection. So one of the technologies that arose from work that we were doing is we've designed a liposome platform for controlled release of these drugs, such that a patient can get an injection about once every 18 months and have a therapeutic level of the drug maintained within the eye in between injections. We also incorporate some lead molecules that we have designed that are very good anti-inflammatory agents. If you would go to the next slide. So in order to move this technology forward, we have created a university startup. This was in collaboration with my business partner, who's also my former PhD student, Dr. Kalyan Karamanchi. And we were really fortunate to be able to participate in Matter and Innovation to Page, two venture accelerators that helped us put this startup together, and also with a lot of support from NIU's College of Business to move some of our technologies into the market. So we have 4 areas that we're primarily interested in, the liposomal drug delivery being the most mature of those technologies. If you go to the next slide, please. As part of working on the drug delivery systems, I had to buy an instrument from Shimatsu Scientific Instruments, and that started a wonderful relationship, which is now allowed NIU to construct and open a core facility, which is a research resource for all researchers at NIU, and partners in the Northern Illinois region, where Shimatsu Scientific Instruments has formed a partnership with us to bring state-of-the-art instrumentation and make it available to the scientific community. This facility is the NIU Molecular Analysis Core, and it's primarily focused on mass spectrometry. We had the grand opening last fall, and we have tools in that facility for analyzing anything from very small molecules up to large biopolymers, complexes, and also carry out imaging studies. If you'll go to the next slide. Okay, my alarm is going off too. Whoop, telling me I have to stop. So once the MAC facility was opened, I'm the director. We have a lab manager, and we're growing our user group. I've now been able to move back and think about my own research again. And this facility has allowed me to really expand and build a number of the diagnostic tests that we've developed. We are currently expanding and building in a number of areas, age-related macular degeneration and diabetic retinopathy, but also we're able to develop some new tests for things like inflammation, which is critical in these ocular diseases. We're involved in diagnostic methods for diagnosing the coronavirus and other viruses, and also cancer with a particular interest in ocular melanoma. We envision these tests to be rapid, low-cost tests that would allow people to have more frequent diagnostic testing with the hopes of capturing diseases earlier, which always has a greater benefit for the patient. So on the final slide, I just have a lot of thank yous to everybody who's worked with me, grad students, the lab manager, my business partner, and then I collaborate with several other industries that I've listed on this slide. So that wraps it up for me. Thank you.
Great, thank you so much, Professor Gaylord. So next up is Professor Boyd Goodson from Southern Illinois University, Carbondale.
All right, thanks a lot, Matt. I'm happy to talk to everyone a little bit about work that we're doing to enhance magnetic resonance imaging with SABR and lasers. Next slide. So as we all know, MRI provides unrivaled imaging of the human body, but it's actually expensive, slow, and lacks the sensitivity to visualize many health indicators. Slide, please. So why is MRI sensitivity so low? Well, MRI often uses contrast agents to enhance the signals from tissues, mostly from water. And the way it works in this case is that tumors or abnormalities, other abnormalities will absorb the contrast agents differentially from healthy tissue, and that gives rise to either bright or dark spots in the images that doctors can then interpret. But the problem is that that agents and our tissues just aren't very magnetic. We can mitigate this problem by using very strong magnets in our MRI scanners, but these are expensive, bulky, confining, and the scanning times are often very slow. Also, MRI is limited to mapping high concentration species in the body like water. Next slide, please. So what we're trying to do is to combat this low MRI sensitivity problem with hyperpolarization. So hyperpolarization is something that we can do to increase the magnetism of these contrast agents by thousands of times. Such hyperpolarized contrast agents can be orders of magnitude more sensitive than today's state of the art. Our technology harnesses hyperpolarization to safely enhance MRI signals to give high resolution without using a super strong magnet necessarily. That can give rise to low cost, very high scanning speed, including even just seconds for full 3D rendering as opposed to tens of minutes. And that, of course, translates not only to better patient throughput, but it also makes MRI amenable to more patients. So on the right, you see an example of hyperpolarized metronidazole. This is an FDA-approved drug that we believe will be useful not only for hypoxia sensing, but hypoxia imaging in the body. Next slide, please. So the idea here is to perform what's called molecular imaging, but with hyperpolarized agents. So molecular imaging simply means imaging or normal imaging of the body, but where signal is encoded from some kind of molecular biomarker for disease. You can currently do molecular imaging with positron emission tomography or PET, but this uses ionizing radiation and the agents are limited in terms of what they can tell you. It's also an expensive technique that takes a long time to perform scans. So we're envisioning instead to do molecular imaging, but with MRI enabled through hyperpolarization. And we expect that we would be able to perform these scans in seconds. This would allow highlighting of disease states, but without the use of any ionizing radiation, no heavy metal agents, providing improved information content over what you would get in PET. And that would also have be repeatable so that you would have fast rescanning times. We expect that this would mean that you would be able to essentially watch the patient's response to treatment almost in real time. Next slide, please. So there's other hyperpolarization technique that exists, but the main technology out there called DNP has its own limitations, namely high cost and low agent throughput. The approaches that we're developing have much more rapid, cost-effective aspects, and they're also scalable. One of these technologies is called SABRE. It's an acronym for Signal Amplification by Reversible Exchange. And SABRE, the way it works is to use a catalyst, which is shown in the picture, to transfer magnetization from a special molecular source, in this case it's parahydrogen gas, to some contrast agent under special conditions that we can control in the lab. And the bottom line is that our SABRE tech can help make hyperpolarized contrast agents that are in water with purity. So we've actually removed the catalyst before it would be administered. The agent is able to maintain the high magnetization and yet have much longer hyperpolarization lifetimes. Next slide, please. Therefore, we think that this also has great commercial potential as well, and that's because the technology is fast, low cost, and enables rapid scans with low-cost imagers as well as conventional MRI scanners. That's why we believe the technology will ultimately be of interest not just to large hospitals where research is done, but also small hospitals, clinics, and even field hospitals with the potential for expansion in developing countries as well. There's, of course, a huge potential market in MRI with roughly 40 million scans done per year just in the US. And by the way, lower costs could, of course, expand both US and international markets. Next slide, please. So where are we with this? Well, with SABRE, so far, we've already been able to demonstrate hyperpolarization in a variety of agents with very high magnetization, long agent lifetime, so hyperpolarization lifetimes of up to 10 minutes, and then sensitivity to various biomarkers of interest, be they for pH or hypoxia or variations in metabolism, biomarkers that, of course, would be very interesting for imaging cancer in response to treatment in various cancers. We've demonstrated hyperpolarization in a variety of safe agents, including metabolites, drugs, and vitamins. And then we've also demonstrated catalyst separation to make the pure agents after the fact. Towards those ends, we've been awarded 2 full patents, and there's 4 more pending or provisional stages with not just from our lab, but our collaborators at Southern Illinois University at Wayne State University. Next slide, please. Before I go, I'd also just like to briefly mention another technology that we're pursuing, and that is to make xenon hyperpolarized with high power lasers. So with hyperpolarized xenon, the idea that we would make the agent and then separate the gas from the polarizing device so that it could then be administered to a patient. So for example, you see there in the false color image, a human subject after they've inhaled hyperpolarized xenon from a polarizer device and then used to perform ultra-fast MRI. This technology development is being championed by a former student of mine, Peter Nicolau, who's also started a startup company called Zeus Technologies, and they make hyperpolarization devices that can actually be purchased like the one you see in the picture at the left. Next slide, please. So that's basically it. With that, I'd like to thank our various collaborators at the institutions you see there, our funding agencies, including NSF, NIH, DoD, and Research Corporation. And basically to let everyone know that if you're interested in hearing more about the hyperpolarization technology that I've discussed today, or if you have any questions, please feel free to email me at bgoodson@chem.siu.edu. Thank you very much for your time and for the invitation to speak here today.
Great, thank you so much, Professor Goodson. That's really interesting stuff. Next up is Professor Johnny He from Rosalind Franklin University.
Yes, thank you, Matt. Hello, everyone. It's great to be here. I condensed everything to one slide, so I want to be sure I can finish on time. So it's a great honor to be with you all and with such a distinguished group of scientists. For that, my sincere thanks to Matt and to the ISTC. I actually started my career in Midwest, not far away from Chicago, Indiana University School of Medicine. Then I moved to Texas, University of North Texas. Then I moved back to Midwest last December. I joined Rosalind Franklin last December. I'm a basic scientist by training. I consider myself as a molecular biologist. My main research interests are in two areas. One is HIV, the other is cancer. As many of you know, it's been more than close to 5 decades since HIV was initially discovered, but there is still much to learn about the virus, particularly the virus infection of the central nervous system, the brain. The reason being, as you know, the antiviral therapy has revolutionized the treatment for HIV-infected population, has improved the quality of the life of the HIV-infected people, but also prolonged their survival. However, very few drugs can get into the brain, which makes the brain a safe harbor for the virus. Certainly cause a variety of the brain problems, particularly now in the antiviral therapy. More than 50% of HIV people display or experience, a variety of the problems. For example, impaired memory, cognitive motor dysfunction, anxiety, or depression. So what we are interested in, or what we have been working on, is to look at how the virus infects the brain cells, how the infection affects the brain cells, and how it causes the brain diseases. Of course, our ultimate goal is to develop therapeutics for this, again, for this very unique situation for which we haven't got a really cure this even so far. So one of the achievement I think we have done from the last 20 years or so, we have developed a very popular animal model. It's a small rodent model to model the brain disease. The HIV infection causes the brain diseases. So this model has been distributed to more than 70 research laboratories and 6 other countries in the world. It has contributed to the therapeutics development, validation, or even the preclinical trial study. The other area of our research is about the HIV infection and the substance abuse. As I said, with the antiviral therapy, the HIV infected people, they live longer, and they have higher quality of life, but it come with that, there are more health problems such as diabetes, cardiovascular, respiratory, or liver disease. Also, many more HIV infected people experience substance abuse, such as smoking, methamphetamine abuse, or alcohol abuse, or cocaine abuse. So that's the other area of our research in the laboratory. Again, our goal is to look at how the virus, the HIV infected people, how the brain, the disease occur in the context of those substance abuse, and try to figure out the intervention for those comorbidity of the HIV infection associated morbidity. The last area, or the second major area of our study is on the cancers. Two main cancers we're working on. One is melanoma, the other one is glioma. Two very, very important, probably most invasive cancers. So this track traces back to more than 20 years ago when our lab cloned a gene. This is before the whole human genome was sequenced. We cloned a gene which we initially found to be very important for HIV, the virus propagation. Later, we found this gene is very important for the tumor invasion and the metastasize for both melanoma and glioma. So we are currently pursuing the possibility of using this gene as a biomarker for those two cancers. So that's all from my research. As I said, I just joined the Rosalind Franklin University last December. I do want to give a shout out to my center, Center for Cancer Cell Biology, Immunology, and Infection. We have quite a number of scientists working in the center, cover from immunology, virology, parasitology, and the cancers. Certainly, the translational research has been one of our priority, not only for our current faculty, but also for the future faculty recruits. Lastly, I do want to thank, our research has been funded by National Institute of Health for the last 20 years or so. So thank you very much.
Great. Thank you, Professor He, and we're certainly glad to have you in the state and at Rosalind Franklin, so thank you.
Matt, can I just add, Dr. He, when I started in nursing, I was an oncology and hematology nurse at the beginning of the AIDS crisis and worked through that. So this is really personal to me, and thank you for your work.
That's great. Nice to hear. Thank you.
Great. So next up is Professor Kathy Nagler from the University of Chicago.
Thank you. I'm delighted to be here today. Can I have the next slide?
Sure.
And the next slide. So I've been studying food allergies for the past 20 years. And during that time, I've seen this disease reach epidemic proportions in real time. I'm sure many of you either have a food allergy or know someone who has a food allergy. And that's because, as I show you here on this slide, this is data from last year, 32 million Americans currently have food allergies. That's 1 in 10 adults, 1 in 13 children. About 50% of each will experience a severe reaction. Food allergies can be life-threatening. And that's a 377% increase between 2007 and 2016. This number is based on physician-diagnosed insurance claims, not self-reporting. So this is a huge change. When I was a child, my brothers and I ate peanut butter and jelly for lunch. By the time my own children, who are now 24 and 28, graduated from high school, their classrooms were absolutely peanut-free because so many children in the classrooms had life-threatening peanut allergies. So how do we account for this kind of change in one generation? Next slide. So we've focused our attention on the microbiome. That is the microbes, and when I say microbes here, I'm referring primarily to bacteria, although there are many other microbes that colonize us, and they're present on our skin and all of the surfaces, mucosal surfaces that are in contact with the outside world. So our hypothesis has been that modern industrialized lifestyle factors have triggered shifts in these bacteria that populate us. And some of those factors are depicted here. Antibiotic misuse and overuse, the consumption of diets low in fiber, high in fat, processed foods, our transition, we acquire our microbes from our environment, and we've transitioned from living in rural settings and settings more in communication with nature to sealed buildings in some cases. We've reduced our exposure to infectious disease, or at least up until the last few months, by vaccination. And we've eradicated previously common pathogens. All of these factors, combined with cesarean birth and formula feeding, have together impacted our microbiome in a way that has depleted populations of bacteria protective against non-communicable diseases, and food allergy is one of those. But I could easily put on this slide other diseases that are increasing in parallel, like autism, obesity, inflammatory bowel disease, and others that I'm not naming now. Next slide. So in my academic lab, we work to identify what these allergy protective bacteria are. We've been able to identify them at both the class and species level. They're now referred to as the gut's peacekeepers. And what these bacteria do is maintain the mucus layer above the epithelial surface that you see here on the left-hand side of your graph. This intact mucus layer forms an impenetrable barrier, keeps the bacteria, food, toxins from breaching the wall and gaining access to the rest of the body. One of the ways this barrier is maintained is by the fermentation of dietary fiber. We can't process the fiber that we take in at all. It's fermented by the bacteria in our guts. And in that process, they produce very important small molecules. One of these is butyrate, which is critical not only for the metabolism of the epithelial lining, but for a myriad of immunomodulatory functions that are very important to our health. In the absence of these bacteria, we can't ferment this fiber, we can't maintain this bacteria, and we have a breach in the barrier. And in this case, food allergens are able to gain access to the bloodstream and elicit an allergic response. Next slide. So what we've done is carry this academic work to real world impact by founding a startup company, ClostraBio. My co-founder is also a 2020 IST researcher to know, that's Jeffrey Hubble. He's the Eugene Bell Professor of Tissue Engineering at the Pritzker School of Engineering at the University of Chicago. And we have together created a synthetic polymer that stably delivers bioactive butyrate to all parts of the gastrointestinal tract as a drug to treat food allergies. Working together with John Flavin, first at the Polsky Center at the University of Chicago, and now at Portal Innovations, we're currently negotiating Series A funding, completing an IND application in preparation for our first in human clinical trials. So thank you today for this recognition as a 2020 IST researcher to know.
Great, thank you, Professor Nagler, and congratulations on all your success with ClostraBio, and good to see John's face again. He's a former board member of ISTC, so many familiar faces in that group. So next up is Professor Leon Shaw from the Illinois Institute of Technology.
Okay, first of all, I'd like to thank Illinois Science and Technology Coalition for the recognition. This honor is outcome of group efforts with strong support from Illinois Institute of Technology. My group mainly focus on advanced material synthesis and processing for energy storage. In particularly, we focus on lithium-ion battery, sodium-ion battery, redox flow battery, and hydrogen storage materials. Today, I think I can share the progress of silicon anode materials with you, given the time we have. Next slide, please. Silicon has a great potential to serve as a high specific energy anode material for lithium-ion battery. At the lower right corner, you can see the silicon symbol there. Silicon have a specific capacity of 4,000 milliamp hour per gram, theoretically. And the lower left corner, you can see the graphite there. It has a specific capacity of 350 milligram per hour. So if we can replace graphite by the silicon, then theoretically we can improve the energy density of the battery by a factor of 10. Next slide, please. However, silicon faces many challenges. In particular, silicon during the initiation and deinitiation, volume expansion by about 300%. Silicon also had problems with a low intrinsic electrical conductivity. And also there's another problem called instability of solid electrolyte interface. So you really needed to pay attention to all these challenges, otherwise silicon will not work. So the diagram below show you the example come from my lab. If you just take a silicon nanoparticles and then put it into anode as a battery, you can see the cycle stability is very poor, which is shown by dark triangles. And you can see that just 20 cycles, capacity dropped by 50%. However, if you coat the silicon nanoparticle with a carbon coatings, and then you can see the capacity retention become better, as shown by red lines. If you introduce the voids inside the silicon particle, which is shown by blue lines, and you can see that now cycle stability improves dramatically. So the idea here is you need a combined nano-sized particle with a conductive network and introduce engineered voids. If you combine all these three, then you could make a silicon anode to work. Next slide, please. So here shows our approach, how to make these engineered particles. We start with a micron-sized silicon particle and do the high energy ball milling, and you end up with a nano-sized particle. And then you do the carbon coating to address the low conductivity issues. And then after carbon coating, we do the sodium hydroxide etching to introduce the engineered voids in it. So we address the volume expansion process, the problem. So the entire process is starting with a low-cost material, and then we have an eco-friendly and industrially scalable process. So basically, it's a low-cost material with very high performance data, which I'm going to share with you. As a result, we filed patent applications for this technology. Next slide, please. So here show you the cycling data. Within 100 cycles, if we have a current density 1 amp per gram, you can see the specific capacity shown by the green line. It's about 1,300 milliamp per gram capacities, and which is 3 times higher than the graphite anodes. And all the cell phone right now, battery is use graphite anode. So you can see that if you're using our silicon anode material, we basically could triple the energy density. Next slide, please. We also done a very long-term cycle testing for our material. Here we show you 1,000 cycles. And in this test, we using extremely high current. So we basically could charge our battery in 6 minutes and with 1,000 cycles. After 1,000 cycles, our specific capacity is still higher than graphite anodes. So you can see that if we can replace a graphite anode with our silicon material, then we can charge it faster and also with a higher energy capacities. And right now we still work on the improve the capacity decay. As you can see, that 1,000 cycle, it decays slightly. So we like to improve that. And hopefully one day, every one of you can purchase the cell phone powered by IIT battery. Thank you for your attention.
Great, thank you, Professor Shaw. I'd love to purchase an IIT battery. That would be cool.
Thank you.
All right, so next up is Professor Andreas Vidal-Gadea from Illinois State University.
Hello, everyone, and I would like to start by thanking you for this great honor, but also thanking the people who made it possible to be here, including Illinois State University and their fantastic support and the students in my lab. So my lab is working on the molecular basis and genetic basis of behavior. And so if you will advance to the next slide, humans, like all animals, they need to interact with their environment. They need to find food and avoid danger and find love. And to do that, next slide, we get by with about 20,000 genes, billions of neurons, and hundreds of muscles. And their job, if you will advance to the next slide, is to basically detect important information and integrate it, and then generate some kind of motor output or behavior. And this is what my lab is really interested in. Now, you can see that, to do this with humans and the complexity of the brain and of the behaviors makes it very challenging, which is why our lab elected to work on a smaller animal that is more amenable to research. Advance to the next slide. And that is the nematode C. elegans, which, while having still the same number of genes as we do, they have fewer neurons and muscles, but they still do the same things. They had to find information in their environment, integrate it, and then produce behavior. They also have a lot of other amenable reasons that make them great for research, but I will point two of them. One of them is that they are easy to work with, and that means everyone can work with C. elegans. Everyone in this meeting, high school students, anyone can be a contributing member of the scientific community working with these worms. And so we've chosen to work on Duchenne muscular dystrophy. It's a mutation. It's caused by a mutation that affects the largest gene in the genome. 1 in 3,000 boys is born with this disease, and it leads to loss of muscle and eventual death. There is no cure for muscular dystrophy, and notably, all the animals that one can mutate the dystrophin gene in do not really get as sick as humans. And so one of the things that my lab had been working on was to develop an animal model of the disease where the animal got just as sick as the humans, and they could recapitulate the disease. And so we want to use these to look at the different steps that link the loss of dystrophin to muscle death. And that allows us to find steps where we might be able to intervene with different treatments. And so you can see the eggs in that picture. The worms develop outside of the animal, so we can watch the muscles develop. We can watch the different structures in the muscle go on, whether they're dystrophic or healthy. And then through robotic approaches that we have developed some high throughput systems that allow us to test thousands of compounds. And we can also create animals where we can watch the muscles contract and measure the degeneration. And so we have used these to look at answer different questions, including evaluating whether different exercise treatments may be beneficial, as in the top there, or we can also have done forward and genetic screens to identify targets for potential treatment. And we are presently in the middle of developing some non-invasive treatments, specifically because my lab is also looking at the effect of magnetic fields and biological systems to look for potential treatments in this muscular dystrophy. Now, because it's easy to work with, thank you. So eventually we also want to use these animals to make copies of the mutations in patients so that we can basically introduce the same mutation as in individual patients into our animals, and then bring to bear this in-depth and high throughput approach to assess different potential treatments, and then feed this information back to physicians so that they can evaluate the potential among all the different options that they have, which may be the most promising one. And as I mentioned, this is done by students. And so over the last 5 years, I had my lab at Illinois State University. I have the pleasure to introduce to science and to research to over 150 students, many high school students, about 70 high school students, but then grad students and even postdocs that had a chance to participate in this research and through different partnerships with the community through different organizations. So once again, I tried to make this as fast, as quick as I could, but I want to really thank my institution for the never-ending support that they gave us, and also to our students and to you for your attention and for this great honor. It was a great honor when I got the award, and after hearing these talks, it's only even a greater honor to see the amazing scientists here. Thank you very much.
Great. Well, thank you and congratulations. Okay, so next up is Professor Tao Zhu from Northern Illinois University.
Thank you. Hello, everyone. I'm Tao Zhu from Department of Chemistry and Biochemistry at Northern Illinois University. Yeah, this first slide pretty much describes the motivation of the research focus in my group. We intend to tackle the global challenge in carbon cycle. Excessive CO2 emission from the use of fossil fuels may lead to adverse changes to global ecological and environmental systems. So a potential solution to mitigate this issue is to intercept part of the CO2 emission and convert it back to fuels, just as indicated by that green arrow. This is a quite challenging energy uphill and entropy decreasing process. So to achieve it, we have to combat two thermodynamical hurdles. First, we should use clean and renewable energy instead of energy from fossil fuels again to power this endothermic reaction, such as electrical energy from solar and wind. Second, we need to combat the entropy by tunneling through the reaction kinetic barrier using catalysts. For all these reasons, my research focuses mainly on the solar energy and catalysis. Next slide, please. Next slide. Yeah, so recently there's a rising star in solar energy field. It's the so-called perovskite solar cells. People get so excited about it because of its fast-growing power conversion efficiency that has reached the level of silicon-based solar cells. Plus, because of its simple fabrication method, in contrast to the very complicated silicon solar cells, perovskite solar cells has aroused a big wave of research effort, becoming a perovskite fever. However, the reality is always brutal. After rock climbing of the efficiency cliff, the field is still facing three big challenges: toxicity, stability, and scalability. We are tackling all these challenges. For example, perovskite solar cells use toxic lead that are fairly water soluble, but without lead, the efficiency drops to the level of no commercial interest. Since solar cells can inevitably get damaged by natural harshness during their application, lead leakage to environment is unacceptable. Especially for this building integrated photovoltaics. Recently, we demonstrate that most of the lead content can be chemically absorbed by putting some off-the-shelf lead absorbing materials right on both sides of the devices. We select a transparent lead absorbing material for the front side so that the efficiency of the device does not deteriorate under normal operation. If the device is damaged, the lead absorbing films automatically and effectively capture most of the water-leached lead content. Next slide. Next slide. Here I'd like to share our more recent work on developing highly efficient electrocatalysts for CO2 to fuel conversion through electrochemistry. We've developed a unique method to produce very clean and atomically dispersed metal catalysts, such as copper single atoms supported on carbon, by using molten lithium as a solvent. And these catalysts demonstrated a record high, over 90% selectivity, or the so-called Faraday efficiency, in converting CO2 to ethanol, with a record low turn-on potential of only 0.4 watts. We also discovered a stunning catalytic mechanism. Under reductive electrochemical potential, 3 individual copper atoms bounced into a 3-atom cluster on which CO2 and water molecules are splitted and reassembled to ethanol molecules. Just as amazing as the water ballet. Since ethanol is the second most produced commodity chemical, and most of our gasoline contains ethanol, this result is very encouraging for our future sustainable energy infrastructure. Very interesting, quite a few spirits and beverage companies also contacted us, besides some well-known industry companies. Next slide. Over the years, my students have worked so hard on different projects in my group. But regardless of their diverse background, one thing in common about them is that they all settled down on a STEM-related career after graduation. So my collaborator also provided great mentorship and opportunities to our students. Certainly without the funding providers such as NSF, DOE, and NARDA, we cannot achieve this. And finally, thank you very much for this opportunity to present our work.
Great, thank you, Professor Zhu. Okay, next up is Professor Jeremy Emil Rosenkranz from Rosalind Franklin.
All right, thank you very much. It's a real pleasure to be here, and I wish I was, I don't know, 20 some years younger so I can actually be in a lot of these labs at this point. This is some amazing science. What I'm going to tell you about very briefly today is something that I'm just a small part of at our university. And I think we all at this point know how common psychiatric and neurological disorders are, as well as the huge economic, as well as the social and personal impact. Next slide, please. And what we've been trying to do at Rosalind Franklin University is to initiate a brain science institute that includes a large number of basic neuroscience researchers that are able to start research or continue research that can lead to very solid, very usable approaches for therapeutics or other types of interventions for patients. And one of the things that we're hoping to do is just to leverage some of the strengths of Rosalind Franklin as a smaller university, as well as some of the flexibility to pursue a whole range of different partnerships and collaborations that are outside the usual types of collaborations with NIH and things like that. Next slide. And our lab's just one small part of that where we're trying to understand the circuits of mood regulation. And we want to understand how these circuits become abnormal in conditions related to stress, as well as illnesses, and why, for instance, our adolescent kids are really bad at mood regulating, and how that might be able to develop as they get older. Next slide, please. And of course, I get to take a lot of the credit for it, but this was done by a whole range of excellent undergrad, grad, and postdocs that are in our lab now and have been in our lab in the past, as well as generous funding. And also, thank you again very much to Illinois Science and Technology Coalition. This is a fantastic honor. Thank you.
Great, thanks, Professor Rosenkranz, and congrats as well to your lab mates here. Okay, so next up is Professor Jean-Luc Aito from the Illinois Institute of Technology.
Hi, Matt and Colin. Thank you for putting all this together, and I'm very delighted to be here, to be among the newly selected scientists to know by the ISTC. Just want to briefly show you what my lab has been doing since 2016. I've been at IIT for almost 5 years now. And so what we do a lot of synthetic organic chemistry. We make new light harvesting chromophores, and we study the photochemistry and photophysics. Next slide, please. So in the long run, what we're trying to do is to develop organic nanomaterials that can be dispersed in organic photovoltaic devices, thereby up converting low energy photon from near infrared into the spectral region where most OPV devices work. And so one other thing we also are trying to do in my lab is to engineer organic polymeric material and use them as drug delivery vehicles. And at the fundamental level, we are trying to understand, how those material can, what is the photostability of those material. Next slide, please. Next slide. So, we developed new organic triple chromophores. And so the question is, why do we care most about triple chromophore? So as you can see here, triple chromophore can be used in synthetic organic chemistry. It can be used as a photocatalyst or photosensitizer to produce valuable chemicals. Triple chromophore can also be used in photodynamic therapy, or can be used also to produce solar fuels. Triple chromophore can also be used to enhance the quantum efficiency of light emitting diodes and also photovoltaic devices. Next slide. So how we develop or design our organic triple chromophore. So by the way, all of you must know that organic materials are lightweight. They can be easily tuned and functionalized. So when we started back in 2016, we decided to focus on purely organic triple chromophore by perturbing the aromaticity of small organic molecule and then recover that aromaticity in the excited state. And in the excited state, we aim to produce 100% of triplet chromophore. So the spin multiplicity also has to come into play. So in the excited state, once this compound absorb light, they can go to the single excited state or triple excited state. So we perturb the ground state with aromaticity, and then we recover that aromaticity in the excited state, and that which will be mostly triple chromophore. Next slide, please. So we well known to use established techniques in synthetic organic chemistry, and we have developed new organic reactions. And I'm just showing you one example here how we are able to design new compound. This is a gram scale synthesis, and we have been able to characterize our chromophore using XRD techniques and also a few advanced spectroscopy techniques. Next slides. So here, the reason I'm showing this slide is to show you that, this is, let's say, one type of characterization of one techniques we use to characterize our chromophores. And this experiment were performed at the Center for Nanoscale Material at Argonne National Lab. And this is where collaboration is very useful for us here in the state of Illinois. So I'm showing you the photophysical fingerprints of these triple chromophores, and we had used from very simple spectroscopy techniques to advanced spectroscopy techniques to map out the photophysical landscape of this compound. Next slide. So as a proof of concept, we were able to use our first generation triple chromophore to as a light harvesting material, harvesting green photons, and we were able to sensitize simple polyaromatic hydrocarbons, and so as you can see, we were able to convert green photon into blue photons. Why this is important? This is important also in biology, as all of you know. Blue and UV photon cannot penetrate our flesh, cannot go through our flesh. So if you are able to harvest red and green photons and up converting those photons in vivo, we can, we will be able to, use the blue photon or our UV photon for, some other type of applications such as bioimaging. Next slide. Recently we tried to devise donor acceptor bichromophoric material, which we plan to study more and see whether we can form solid crystalline materials of this compound. And so we are still studying this type of compound. And next slide, please. And we were able to synthesize new other type of acceptor bearing biomolecules. So here we are showing that we can perform the energy up conversion also in biomembrane. So having a cholesterol moiety on the acceptor moiety there, you can see that we are able to convert green photon into blue photons. Next slide. So just a highlight on the other project from my group. So we are trying to use these small molecular building blocks to engineer two-dimensional organic polymer. And so we want to see whether we can deliver carbon monoxide in vivo. And as you can see, some of these compounds, have like a carbon oxygen double bond. And so if you can release carbon monoxide in vivo, that would be great. So at the fundamental level, what we are trying to do also is to understand, if we photolight this material, what will be the type of, light-induced chain, if we can see light-induced chain propagation within the frameworks of these materials. Next slide. So I'm, showing this slide just, to show you the picture of my group. That was last summer. And also showing you some of undergraduate students, my undergraduate students who are involved in the two projects we have in my lab, and some other outreach programs I have developed or initiated here since 2016, and by training high school students and underrepresented minority students. Next slide. I sincerely want to thank my students for for this work, and I think they should be they are the start of the lab, and without them I don't know if we should be able to do all these things. And I also want to thank my collaborators and also the National Science Foundation and Illinois Institute of Technology for funding this research. And thank you for your attention. Thank you.
Great, thank you, Professor Aito, and congratulations, especially on your K-12 work. That's really cool to see.
Thank you.
So with that, I want to thank all of our researchers for presenting today and congratulate them on being Researchers to Know in 2020. I also want to thank all of the university administrations that worked on selecting these researchers with us. Because this isn't just something that we make up, right? We work with leadership at the universities to identify really kind of the stars of research each year. So, I think that's what makes our Researchers to Know honor so special. So thank you to all the researchers for presenting today, and to all of our partners at the universities across the state for working with us on this. We are a little bit over time, so unfortunately we don't have time for questions. But we will be sending around a follow-up email after the event to all of our RSVPs. So if you have questions, feel free to respond to that email, and we'd be happy to answer those. Or if you'd like to be connected to one of our researchers here, you can, again, respond to that email, and we can make those connections. So with that, yes.
I thought somebody promised donuts, but that's okay. Maybe next time.
Yeah, well, yeah, maybe when we're in person. Digital donuts for now, unfortunately. But yeah.
Great crowd. I enjoyed it, Matt. It's my congratulations to all of you.
Great. Well, thank you, everyone, for participating.
Thank you, everyone.
And thank you to all the participants for being here today. And I think a big round of applause to Matt. You did an amazing job.
Well, thank you. Thank you. And thanks to Edith as well. She's really been amazing putting all of this together. So big thanks to Edith as well. So hopefully in 2021, we'll be together and we can share donuts and coffee and say hello. So
Spike donut, my recommendations.
Yeah, there you go. That sounds good to me. All right. Well, thank you, everyone. And until next time, take care.
Stay healthy, everyone. Thank you.
All right.
All right.
Stay
bye.
safe, everybody.